Hello, I'm Tim Wall, Senior Reporter for Pet Food Industry. Thank you for joining us for Ask the Pet Food Pro. And in this edition, we'll be discussing pet health from the inside out, the microbe's expanding role. Ask the Pet Food Pro connects experts with industry professionals and offers the opportunity to pose a question directly. Ask the Pet Food Pro is brought to you by Pet Food Forum and Pet Food Industry's journalists and editors. You can find our reporting and analysis at petfoodindustry.com. And you can subscribe to our daily newsletter and monthly magazine. Today's discussion focuses on the hidden world of the gut microbiome and how it influences pet health and wellness. Pet food ingredient producer Cargill sponsors this edition of Ask the Pet Food Pro, and our expert guests are Dr. Mark Franklin, Global Technical Lead for Cargill, and Dr. Jessica Jarrett, R&D Principal Scientist for Pet and Lifestyle at Cargill. Welcome, Mark and Jessica, and thank you for joining us today.
It's great to be here. I'll introduce myself. I'm Jess Jarrett, leading up global R&D for pet at Cargill. My scientific background is primarily as a microbiologist and a computational biologist as well. I've worked with host-associated microbiomes from a pretty wide variety of hosts and environments, and I've been in the pet microbiome space specifically for about 7 years now. On a personal level, I'll also say that helping pets is a big interest of mine and have spent a lot of time volunteering as an adoption counselor at some local cat rescues.
And I'm Mark Franklin, Global Tech Lead for Pet with Cargill. Happy to be here today, Tim. Thanks for the opportunity. So my background is in pet food formulation, product development, nutrition. I'm an animal nutritionist by training and I get to work with lovely people like Jess. Looking forward to today. Thanks.
All right, but before we do get started with that discussion, I do have a few announcements. Audience members, you have all been muted to ensure that everyone can hear and focus on the discussion. I've included many of the questions that you all sent in before into our script, but if you do have questions during the discussion, please submit those through or any other comments to the chat box. Please note that this chat is being recorded. You can access the recording starting in about a week on the Pet Food Forum events website. So now, when we're considering the microbiome's role in pet health from the inside out, we often think of the gut in terms of digestion, but how does the gut microbiome in dogs and cats go beyond digestion?
Yeah, absolutely. A huge part of the immune system is located in the gut. Bacteria and microbes in the gut can produce a lot of different metabolites, especially things like short-chain fatty acids, things like butyrate. And those molecules can really help to regulate the immune system, to promote appropriate tolerance to the good bacteria in the gut, as well as contributing to gut barrier integrity and protecting the body from harmful pathogens, from other types of molecules that shouldn't be entering the bloodstream, basically, if they're too large or too antigenic. So all of that to say, the microbiome can really modulate and gear up that immune system so that the response to environmental challenges is faster and more appropriate. And we do see that response in circulating white blood cell profiles when we have different microbiomes. Also worth saying that in a lot of other species, as well as in pets, there are really strong links between the microbiome and different autoimmune diseases, metabolic syndromes, even things like cancers.
Yeah, and building on that a little bit, looking at the effect of the gut microbiome on other organ systems, right? So we've looked at skin and coat, we've looked at that gut-brain axis, we've looked at that gut-oral axis, really that gut microbiome serving as a master controller and having lots and lots of influence on the rest of the pet's body. One of the intriguing areas that we're just starting to get into is looking at other organ health, like kidney health in cats. So lots of different effects there that gut microbiomes playing a central role in.
Considering all those different realms that the microbiome plays a role in, let's narrow in a little bit and explore a few of them. How does the pet microbiome influence the immune system, their systemic immunity, and what are some of the implications in that sense for preventive health?
Yeah, I think when you look at what's a healthy microbiome, what's a more dysbiotic or imbalanced microbiome, that imbalance has been linked to a lot of different diseases and conditions across many species. So yeah, I think that's really pointing to that importance and to, again, training the immune system appropriately and having an appropriate level of immune response and inflammation systemically through the whole body. Mark, I feel like this is one of your key interest areas too.
Yeah, and for me, the fascinating part of this starts to come when we start looking at the inflammation in other areas, particularly around joints and joint health and mobility, and just really affecting how our dogs are feeling, how they get up and move, how cats are jumping or not jumping in older age, and looking at that effect of immunity, right, as monitored by the gut microbiome, but then having those effects systemically.
Gotcha. And zooming out then from the interior and immune system to looking at kind of the pet as a whole, are there connections between the microbiome and the way a pet behaves?
Yeah, I
Yeah,
would
this is so
huge.
fascinating. I love this research. I think it's, anyway, I'm amazed that people really started looking at this and all the things that they're finding. Caveat that a lot of this work is happening in model systems that are a little bit easier and more tractable. So things like mice, but we are starting to get into this for dogs, not so much for cats, but definitely for dogs. We're starting to get into this. So there's a lot of connections between the gut microbiome and all sorts of behaviors, social behaviors, fear, anxiety, attention, all the way up to learning and cognition. And in dogs, we can see some of these differences there are differences in the microbiomes, right? Between like fearful dogs and non-fearful dogs. And that's tied into differences in the metabolites that are coming from that microbiome. So they can produce neurotransmitters, they can degrade neurotransmitters and produce all these other psychoactive sort of chemicals. So also really interesting that this is a bi-directional influence. So behavior can influence the gut and gut can also influence behavior, which is really interesting. And again, a lot of that mechanistic work is happening in mice. So like one of the papers that kind of came out recently that I found really impressive, you can stress these mice and that will reduce the stimulation of the vagus nerve. And then there'll be less mucin produced by these glands in the gut. And then that mucin being absent is not available to feed specific strains of bacteria. And so then you have this altered gut microbiome just by making your mice feel stressed out. So it goes both ways, which I think is the most amazing part, really.
It is. It's amazing that it connects like that. So you were talking about rats right then, and we have a question come in through the chat about research on different species. So most of the research being done on dogs, cats part of this too. And what are some of those differences that occur between the canine and feline microbiome?
Yeah, I would say writ large research, I would say tends to focus more on dogs. Particularly behavioral research, I would say focuses more on dogs. I can have my own personal feelings about that as a cat person, but also there are certain things that are going to be easier in dogs or in cats, right? Writ large, cats do have a slightly more diverse, but also a more consistent microbiome from individual to individual relative to dogs. A lot of these differences are going to be due to differences in intestinal physiology, the types of foods that we feed cats and dogs, the nutrient profiles of those foods. Both are pretty adaptable. I would say that more of the research, maybe like 70-30, has focused on dogs rather than cats to date. But I do think that's starting to shift as people are seeing more growth in cat ownership and spending on cats.
And the thing that amazes me about that is too, just looking at the adaptability of both dogs and cats to be able to shift that microbiome depending on what food source or what they're eating, and in conjunction with that intestinal physiology, very adaptable.
And the variability, I will say, is huge, right? So like we can see what a sort of healthy dog microbiome looks like, but the variation between individuals is enormous. Even on sometimes exactly the same diet, the variation can be really enormous.
So even in between one dog versus another, and then that same dog eating one diet versus another, it's just very complex.
Yeah.
Yeah.
And Tim, even in dogs, when you start looking at adult body size, the difference between small dog, large dog, even fed same food, get a completely different microbiome.
Young versus old, all that. Yeah. Yeah.
That actually brings up, there was a kind of follow-up question in our chat here that was asking about similarities with human and pet microbiomes. I know there's so much interest in the human realm of gut microbiome. How has that played into interest in the pet microbiome?
Yeah, one of the kind of jokes in the field is that like we are the model species for dogs and cats, which is the opposite of how that usually works, right? Because there is so much more, I think, of financial investment in the human microbiome space, right? In terms of therapeutics, custom approaches, all of that sort of stuff. It is interesting the exchange of microbes between humans and dogs and between dogs and humans and cats as well. For instance, I think sometimes pets will be blamed for spreading bacteria in the home. But when it comes to, for instance, the transfer of antibiotic resistant bacteria, that is often happening from the human to the pet rather than the opposite.
Huh.
Yeah. Which is a
So-
whole other can of worms that we don't necessarily need to open, but yeah.
Yeah, that's a whole nother show right there.
Yeah.
Yeah, let's get back to some of the different ways that the microbiome interacts with systems. Could you explain a little bit about how the gut-brain axis in pets is affected by the microbiome and how that affects cognitive health in pets?
Sure.
Yeah.
Yeah. Just
Oh,
jump.
you go, Mark.
No, jumping on that, I guess from a higher order point of view first, we know that microbiome can produce a lot of those brain altering chemicals. We know that the microbiome degrades a lot of those brain altering chemicals. And as Jess mentioned before, that double feedback loop, right? So the microbiome influencing what's going on in the brain, what chemicals the brain are seeing, and then the brain sending those signals back to the gut and that interplay there. So definitely that link there through the production of those hormones and chemicals. I will say it's a very complex area to start to understand. And so as we look and start trying to understand that and start driving some of those things, there's lots of other factors that come into play. Guess I'll let
Yeah.
you expand.
Yeah. Yeah. Part of why that's so complex is because there are a bunch of different mechanisms by which the microbiome can exert these effects, right? And similarly, the body can exert the effects on the microbiome, right? We've talked about even things as simple as like the production of mucin by various glands, vagal nerve stimulation or lack thereof. Again, yeah, the degradation or production of those neurotransmitters, as well as generalized systemic inflammation, or if gut barrier integrity is compromised, inappropriately letting things cross blood-brain barrier and all of that sort of stuff, and dialing up inflammation systemically in the body can also have a really big impact. There is, again, humans are the model species for pets here, some really interesting work between specific taxa or specific groups of bacteria and cognition, especially in cognitive decline, where we think of inflammation as being a big part of what produces what we think of as aging, and also some really interesting indications that different diets or probiotics might be able to improve cognitive function in people, in mice. This is also something that I think we could test in dogs, because there are controlled behavioral tests where we can see how good dogs are at learning or remembering or figuring out a puzzle. So I think there's opportunity here for some really interesting questions.
Once the testing is done on humans, then let's speak about one of the gut where it all starts. How is the oral microbiome's health and composition related to the gut microbiome? Can it be an indicator? We had a question about this from an audience member before the show.
Yeah, this is super interesting. I think there's a growing appreciation of the importance of oral health for whole body health. This is also linked to cognitive decline and other health issues, let's say, if oral health is compromised. As you say, the mouth is the beginning of the GI tract, right? So microbes can move directly from the mouth into lower parts of the GI tract. And they can also, if tissue in the mouth is compromised, can also be translocated through the bloodstream or through immune cells. So I wouldn't necessarily say that there is like a one-to-one connection between the oral and the gut microbiome. Like I wouldn't necessarily use the oral microbiome as an indicator or a biomarker of what's going on in the gut, but they do communicate with each other. So you can like experimentally add, for instance, periodontal disease associated bacteria to the mouth, and that does have negative effects on the gut microbiome, which is interesting. And yeah, a lot of these things are, again, coming from those same sort of sources of dysbiosis, systemic inflammation, all of that.
And I think we're starting to understand, Tim, a little bit more looking at the oral microbiome and starting to understand that there's several different communities of oral microbiomes, even in the oral cavity, depending on where we're located, gum line or cheek or under tongue, and those different populations exist. And looking at that influence of the gut microbiome, even from a systemic effect, right? Having influence and feedback loops on what's going on in the mouth. So yeah, lots of connection, but as an indicator, as Jess said, it's pretty difficult to use one as an indicator for the other.
So speaking of what would be a good indicator, are there some reliable current methods for analyzing the pet microbiome?
Yeah, I think whatever method is best is going to depend on the question that you're asking. Certainly, I think we've all settled on fecal material as a really good proxy for what's happening in the gut, although I will say it's not necessarily identical to, for instance, microbes that are really tightly associated with the mucus lining of the gut. That can be like a slightly different community. But a lot of the research that we do have is coming from fecal samples. So I think certainly there's a lot of background and context and other results that you can interpret that data in the context of. Those samples are going to be obviously easy to collect. There's a lot of really good preservation options for that. And then once you have those samples in hand, there's a few different ways that you can go about assessing who is there in terms of microbes, as well as what are they doing, if you want to dive deeper into that. So there are a lot of sequencing-based methods where you can either go in a very targeted fashion with something called qPCR, and this is how, for instance, the dysbiosis index works. There's a set of specific bacteria that looks for, and it quantifies them very specifically, whereas other methods are less targeted, and you get a snapshot of kind of all of the bacteria that will be present in that sample. As well, there are other methods like shotgun metagenomics, where you can get a glimpse not just of the identity of the bacteria, but as well their genetic content, so the capability that they have to have certain metabolisms or to produce certain molecules. You can get into that. So I think what you want to do is going to depend on how much money you have, number one, and what your question is. If you want to know about dysbiosis, something like the dysbiosis index can be great and is really consistent and replicable. When you get into untargeted methods, it's good to be mindful of differences between, for instance, different sequencing labs or between details of specific methods, because your results may or may not be comparable to what other people are doing. That's a really complicated answer, which is my sickness as a microbiologist. Mark has dealt with this a lot.
And it's so complex.
Yeah.
And I think it behooves us to look at some of the other species work too, particularly in some of our livestock species, where we've been able to look at the microbiome from individual portions of the gastrointestinal tract. And right, you've got, let's just say you've got a certain bacterial species hanging out, not doing really anything in that part of the tract. But when it gets to a different part of the tract, or similar genus species strain, even in a different part of the tract, has a completely different function and job. And so right now, the fecal microbiome is, in dogs and cats, about the best we can do right now. Really looking for other techniques and other methodologies to start understanding what's perhaps going on a little bit further upstream, maybe into parts of the small intestine, even into the stomach, and seeing how that all interacts and plays. So we're getting a good understanding, but we've got a long way to go to really wrap our arms around this.
Extremely complex. So sounds like a whole little universe in there.
Yeah.
So when we're going from considering all the benefits that we've been looking at of a healthy gut microbiome and how that affects the rest of the pet, what can pet food producers do to encourage that? What are some shelf-stable microbiome targeted ingredients that really show promise, that can really help promote that gut microbiome balance?
Yeah, for sure. Great question. There's a lot of functional raw materials that we can use to influence the microbiome. It really comes down to what we're trying to target and considering the variability that we're dealing with, right? We're all pretty familiar with the biotics, right? Prebiotics, probiotics, postbiotics, symbiotics. We've got these biotics that we can utilize. A lot of time that comes down to what can we get, which biotic can we get to which part of the intestine and still have functionality, right? And each of them have their pros and each of them have their cons. And then when you bring in the shelf stability, okay, is that shelf stability at date of manufacture? Is that shelf stability at end of expiration, end of food life expiration? So a lot of those things have to be considered when we're looking at the biotics. As well, there's some other functional ingredients that we're looking at, other people looking at as well. When you start talking about some of the polyphenols, you start looking at some of the botanical phytogenics, you start looking at carotenoids. There's a whole host of functional ingredients that can be considered, but as importantly, you then have to consider them in pet food manufacturing processing and how we're going to deliver those.
Gotcha. So speaking of that, the possibility that perhaps something that goes in doesn't come back out, how can a pet food manufacturer best validate their microbiome health claims, ensuring that those probiotics make it through the processing and then make it through regulatory and actually market that?
I think when you start talking about claim substantiation, start with the owner in mind, right? What benefit is that pet owner going to see their pet having? We can certainly make microbiome claims that promotes a good microbiome or promotes a more balanced microbiome. I wonder a bit if we're talking to ourselves in the pet food industry about that. Does the owner really care what the microbiome's doing? They want to see no accidents. They want to see firmer stools. They want to see all those things that we started to mention about with the other body axes, less anxiety, skin and coat. And it is a little bit scary there when we start talking about those claims, because we're not going to stray over into those drug claims from a microbiome with those functional additive products. And so really it comes down to those claims that make a difference. When manufacturers are looking for that assurance, then there's really a lot going on inside the gut. And we've already discussed how complex it is. What's the outcome, right? What's the outcome for the animal? And that's where we need to build our substantiation in our claims.
Yeah, to me, that's the starting point, right? Because if you don't have that visible benefit that pet parent can see, that's really where you need to start. And as a scientist, I like to then ladder that back up to what samples can I collect, or how can I analyze this to see how that benefit is being created in the animal, right? So if I'm seeing firmer stools, or I'm seeing like less fecal odor, can I look at metabolite production? Can I look at the microbiome and say, I think that this is why this is happening, and I think this is how that benefit is being produced. And that's, I think, how you bring those pieces together, right? Between what the pet parent cares about and then what we're doing in terms of formulation and in the lab.
Gotcha. And with those different kind of pieces that you're putting together in the lab and the claims that can be made from them, you had mentioned earlier, Martha, prebiotics, probiotics, postbiotics. Each of those has different parameters for when they're being processed, and then also different claims that could be made about them. Could you give us a little kind of overview real quickly of the differences between how prebiotics, probiotics, postbiotics support a healthy gut microbiome?
Yeah, sure. So yeah, lots of conversation around pre, pro, post, sin. How do they interact? What do they do? What's their function? So just really from a high level point of view, right? Prebiotics is that bug food, that good bug food, right? We want those prebiotics to consume by those commensal bacteria in the intestine, and we want those commensal bacteria to produce metabolites. Those metabolites influence their own populations. They influence the environment around them. They influence other microbial populations, as well as some of those metabolites are crossing that intestinal barrier, right? Having effect systemically in the body. So that's the job of prebiotics, right? To act as that food. When we look at probiotic, we're trying to provide those good bugs to eat those prebiotics to make those metabolites, right? Those metabolites, variety of them and various functions. Postbiotics really comes along and says, hey, what if we feed metabolites directly? What if we feed those metabolites to influence the population of the microbiome, to shift those to a more favorable status? I air quote favorable, right? We're still defining favorable. And to have those effects in the rest of the body as they cross over. So really trying to remove some of the variability. Do I have the right prebiotic? Is in the right amount? Do I have the right probiotics? Are they eating what they're supposed to eat? What happens in a stressful environment? What happens when the dog goes through a period of sickness, or the cat's working on some massive hairballs? The variability associated with that. Fascinating when we start thinking about combinations of those, right? Looking at prebiotics and postbiotics, looking at syncs, looking at all the different combinations of those biotics coming together to see, can we have a synergistic effect?
So within that complexity of options between prebiotics, postbiotics, and all that, or even blending them, we had a question from the audience asking about differences between the sources of biotics, whether they're from bacteria or yeast. Is there a difference in functionality depending on the source of the biotic in terms of gut health?
Yeah, Jess, why don't you start on that?
Yeah. There are, I think most of the postbiotics, bacterial postbiotics, I should say, that are out there are, again, coming from different lactic acid bacteria. And that's a lot of what's being produced in those industrial sort of quantities. There are some really interesting postbiotics out there from other types of bacteria. For instance, in the human space, people are looking at postbiotics made from Akkermansia, especially for metabolic health. So I think that I'm very excited about this space, and I think that there's tremendous potential to really explore whole new benefits. So When it comes to bacteria versus yeast, I think a lot of what we need to look at is more so than like the kingdom difference, just looking at those specific strains and what are the benefits, functional benefits tied to that specific strain. Because you can even have, for instance, like different strains of lactobacillus that are going to have different benefits that they're providing. So I think it's helpful to look at it on that really granular level versus the categorical level. Mark, I don't know if you probably have more that you want to say here too.
No, I just was going to add when we start looking at the differences between what bacteria produce and what yeast produce, and then being able to take each of those and start combining those, I think we open up the space even more. And
Yeah.
so looking at what certain bacteria are able to do, a lot of those make it more difficult on us because they're strict anaerobes. And so as we start looking at the benefits of some of those, commercially growing those and then taking those and using them either as a prebiotic or turning them into a postbiotic presents some different challenges. But I certainly think there's some advantages to looking at both of what bacteria and yeast probiotics into postbiotics can provide.
Gotcha.
Yeah.
Yet more tools in the toolbox and complexities. So speaking of actually of different options and complexities, we had another question from the audience that was asking about the actual pet food format, whether canned, fresh, extruded, or freeze-dried. How can that affect the microbiome?
Yeah, this has a huge impact on the microbiome of pets. Worth noting from the start that I think pets on all of those different formats can absolutely have healthy microbiomes, right? It's not that one format is necessarily always better or worse than another. It's about what's going to work for that particular pet and all of those other needs in the space. Also worth noting that because of manufacturing limitations concerns, some of those formats tend to have really different macronutrient composition as well. So if someone is feeding a raw food to their cat, that's by necessity going to have a really different macro composition than if they're feeding a dry kibble to the cat. So we're going to see differences in microbiome composition that occur because of this diet has more protein or this diet has more carbohydrate. And that's totally expected. In terms of cooking, there's some really interesting research here, again, from mice, unfortunately. But what they did was this really simple experiment where they fed mice just meat or just a plant food, which I think was sweet potato. And it was either raw or cooked. So raw meat, cooked meat, raw sweet potato, cooked sweet potato, and looked at the effect on the microbiome. And whether you cook meat does not affect the microbiome, at least in mice. Whether you cook sweet potato matters a lot. That changes the microbiome pretty drastically. So I think that's really pointing to the differences in like the breakdown and digestion of fiber specifically. So I think there's some interesting food for thought there when we're thinking about pet food, right? That it may really not matter if from a microbiome perspective, if you cook or don't cook the meat. So there's some food safety stuff that we could think about there. As well with cats, one of the strongest signals that we see in the microbiome in terms of diet is whether or not the cat eats any dry food. And some of my previous work, I've run like a machine learning classifier on samples. And it's very good at distinguishing cats that eat any dry food from cats that eat no dry food at all. So that's a really interesting signal in cats in particular. But all of the diets can be great. All those formats can be great. They do alter things.
Yeah. And Tim, I'm amazed by the adaptability, right, of the microbiome. So the redundancy that exists in the microbiome and the ability to adapt to different diets and still have what we would consider a good microbiome, regardless of diet format, speaks to me to that grand design of the microbiome in being able for our pets and us as well to adapt to different foodstuffs, food formats.
That kind of ties into a question that we have in the chat asking about how, if there is any kind of standard, what is a good microbiome in cats versus dogs? Is there an ideal? Is there a standard? And what kind of challenges does that make of deciding whether something's in dysbiosis or not?
Yeah, this is the eternal question. So I would say that there is enough variation between individuals, and as Mark is saying as well, enough sort of functional redundancy that I would not say that there's like one specific composition that's necessary, right? We can look at the literature and at the data and see that there are certain taxa that are with very high frequency found in healthy cats or healthy dogs, right? From a simplistic, like a logical perspective, it would seem that those are probably important to have if you want to be a healthy cat or a healthy dog. And if all or most of those particular groups of bacteria are missing, that's maybe something to be concerned about. There's also the work around the dysbiosis index is also really nice. A lot of those are pretty important species, either on the positive or the negative side. So I think there's good functional evidence to show why some of those specific bacteria are important either to have in the terms of something like Clostridium hirinonis, which is like really involved in bile acid conversion, for instance. Without hirinonis, it's anyway, there's an important function that can be missing sometimes in the microbiome. As well, there are certain taxa that when they're overabundant, we know that really tends to come with problems. That's things like really high streptococcus can do that, really high levels of E. coli can do that, especially leading to diarrhea and whatnot. So again, there is a lot of variability, and it can be hard to, you need a lot of samples and a lot of data to extract that insight from the literature, which is unfortunate. There are some companies out there that are working in this space, but much of that stuff is proprietary or belonging to those companies, unfortunately.
I'd add to that saying I think there's many possibilities and definitions of good or stable. Quite obvious when it's bad, at least from a chronic point of view. Some of those are easier to characterize,
Yeah.
particularly for the pet owner. I think the secret sauce here comes in understanding how we could take one that we would classify as okay or good and start to tweak that to be better, right? To optimize some conditions for the animal, for our dogs and cats, as we think about their life stages going all the way from puppy kitten all the way to senior animals and that whole spectrum in between, looking at, okay, we may need to do something different in a young animal versus a healthy adult animal. As that healthy adult animal starts to age, then what do we need to think about? What do we see issues start going wrong? Okay, the microbiome's not bad, but let's understand what's starting to change as that animal goes through a life change. And can we come with an intervention that would provide a benefit?
To follow up on that a little bit, are there opportunities for more like personalized or very condition-specific formulations based on a specific animal's microbiome profile or a particular issue that they might be having?
I love this question. So there's a lot of people spending a lot of money on this question in the human space, I will definitely say, and a lot of really interesting research, particularly in regards to blood glucose control, metabolic health, type 2 diabetes, IBD. There's a lot of energy being put into that. One of the things that you really need to make that work is a lot of data, which is tied back to the individual. And then also ideally that you have some sort of call and response or some sort of feedback that you're getting from that individual. That can look like if you're targeting like blood glucose control, that can look like continuous glucose monitoring while you feed specific foods or test foods or combinations of foods, right? Or it can look like microbiome testing. I don't think we're necessarily there yet with pet data, but I do think that we could get there. There's, again, real interesting stuff in the human microbiome space where you can predict somebody's blood glucose response to specific foods based on their microbiome. So I think there's a lot of room there for us to go to some interesting places with pet. I think there's also some low-hanging fruit in terms of personalization, where if we're seeing specific obvious issues in the microbiome or specific bacteria that are missing, we do have some interventions where we can either, whether that's prebiotics or whether that's trying to add back in some of those beneficial microbes, where we can do that on an individual basis, but it's certainly a growth area for the future.
Gotcha. So continue on with the idea of formulating and how to actually influence the gut microbiome. When pet food formulation teams are attempting to integrate these insights into how the microbiome influences the pet, how can they create that pet food without then compromising its palatability or its processing stability?
Yeah, that's a great question, Tim. So as you look at incorporating some of these functional ingredients into the formulation, palatability becomes key, right? Key concern. Fortunately, many of our postbiotics, including yeast-based postbiotics, have been shown to be super palatable, unlike some of the other functional ingredients that we look at, particularly from a botanical point of view, right? And so if we're going to choose one of those to have a specific job to do, we're going to have to think about, okay, what's got to be true for the cat, for the dog, and or especially the cat in those cases, to consume that. And we can do that in several different ways. Formulators can work around that, including some different palatants, some different maskings. From a processing point of view, you have to back up and say, okay, what functional additive am I using and what is its stability during processing? Okay, if it's not going to make it through retort or extrusion, let's then start focusing on, okay, can I add it post-kill step, right? Can I look at adding that perhaps at a coating step or at a step past that heat kill to drive that effect? Oftentimes that brings along a whole different set of challenges, right, in trying to incorporate those. But it's doable and it can be done. But then really picking those functional ingredients that we know are going to be either PAL positive or PAL neutral, and figuring out where we put those in process to have the desired effect becomes key.
Let's see. I believe we have gotten through most of the questions we had here up to this final one about, so we've been talking a lot about some of the advances that are being made in the science of the gut microbiome, but there's also a lot that isn't known yet. What do both of you consider to be some of the biggest gaps in our microbiome knowledge, and what should be prioritized within the next 5 years or so to really look at?
Mark, do you want to start there? I feel like
Yeah, I'd
my
love to.
natural tendency is more pie in the sky. So you can start us off on planet Earth and I'll take us into outer space probably.
Yeah. For me, Tim, it's about being able to harness the power of that big data, right? As Jess has alluded to many times, there's lots and lots of data that we have to have. I will say that the price of obtaining that data is coming down, so that's a great thing, right? But we've got to have thousands upon thousands of data points and being able to then harness that and work with that in real time, right? It doesn't really do any good if I'm trying to make an intervention and it takes me 6 months to understand what's going on, right? So understanding how to handle that data, I think will be a huge breakthrough in the next 5 years. Now, as we gather more of that data and start understanding that, the interplay between what's going on in the gut, more specifically with those different taxa, looking at how certain taxa affect certain other taxa, how they play together, what are they driving from a response, as we've talked about already, that gut brain axis. So understanding more and more of that, I think, with the collection of that data, and then learning, okay, if I do this, can I influence that? Or did I get absolutely the opposite reaction that I wanted? So figuring out how those populations play together using big data, I think will really start to unlock a lot of understanding for us.
Yeah, my answer is similar as well. Bringing in when we're talking about health effects, right, and understanding what does it really mean when you change the microbiome in this way, bringing in some of this other tech, whether that's like smart cameras or wearables for pets, that other information about how the pet is doing and its health, I think is going to be really critical. Yeah, and trying to get to a place of being predictive rather than being reactive, I think is where I would love to see us be able to get to.
All
And
right.
preventative as well. Oh my gosh. Yeah, there's so many things where you really feel like there have got to be indications in the microbiome probably years ahead of symptoms potentially, where if we could intervene there, I think it would make such a difference for pet health. So
And that would come back to having to analyze all that data, like Dr. Franklin.
exactly.
A lot to be sifted through.
Yeah.
Speaking of sifting through some things, let's take a look at some of the questions that we got now in the chat. I've been trying to bring some of these in as we've been talking. So we have a question here about, given the conditions that a kibble might be stored in a home, unsealed bins, fluctuating temperatures, or exposure to air, how likely is it that probiotics included in these diets will even remain viable over time? And how should consumers then evaluate any packaging claims or probiotic claims?
Yeah, that's a good question. I think that really then for me, that pushes back to the manufacturer to say, okay, can you show me your stability data really in ambient testing, in ambient storage conditions? Okay, the bag's been open and the bag's open, exposure to air, quantify for me, does that matter? Start to understand what that degradation curve looks like. And then the age-old question of how many do we need, right? Is it 10 to the 8th? Is it 10 to the 6th? That depends on the strain, right? That depends on the individual genus species even of that probiotic and what effect does that have? And then, okay, start to understand I might need this number for that effect, but I need a higher number to drive a different effect, right? So really for me, I go back to say, okay, where's the data that says survivability exists across that shelf life. And there are probiotics that will do that, right? We know that. But having that confidence and that for that consumer peace of mind to have that point of data is important.
So speaking of specific data points, we have a question here about information about short chain fatty acids like butyrate as a postbiotic and any research supporting its functionality.
Yeah, the unfortunate thing about butyrate specifically is it's real gross. So, as anybody who's ever worked with it would know. So yeah, butyrate is super important in terms of feeding host cells, and the gut has a ton of research really supporting its importance. It's great if you can get the microbes in the gut to produce it at that site. Microencapsulation in terms of feeding it directly is also something that can work. I know in the equine space, that's something that people do. Yeah. The trouble with some of these things is number one, trying to get them past the stomach sometimes, past the acidity of the stomach, and then also sometimes trying to get the pet to be able to actually eat it if it's objectionable in some way. So sometimes it can be for some of those types of things, although it is challenging to get the microbiome to produce a specific metabolite, sometimes that may be your best bet. Mark, I don't know if there's more you want to say there.
No, exactly, Jess. I agree. The other thing about that volatile fatty acid, short chain fatty acid profile is where do I want it? And how do I, as Jess referenced, how do I get it there? Right? Butyrate is a prized molecular compound for the gastrointestinal tract. And we know that it's going to be sucked up just about as soon as it's made, right? Intestinal cells love it. So how do I then have the effect in the small intestine, particularly the distal parts of the small intestine where, a lot of that digestion absorption is taking place, but then I need intestinal cell health in the colon. And so to be able to feed that directly and hope that it gets to the colon in an unprotected form, probably not going to happen. So we need, as Jess said, look at the microencapsulation potential, or better yet, I would say, how do we influence the native microbiome to drop that volatile fatty acid production in the different parts of the small intestine through either the combination of influencing that microbiome and or adding metabolites that we know stimulate that volatile fatty acid production.
Question here following up on that idea of influencing the microbiome format or the microbiome makeup. How does the canine microbiome shift based on diet format? If you're maybe a little bit, you can go on about how if somebody switched a dog from kibble to a wet, how would that influence their microbiome?
Yeah, again, this is going to partly depend on the macronutrient composition of the food because you can take, so for instance, there's lots of great research where you can take the same diet format, but alter the macronutrient composition and produce microbiome changes in that way, right? So when we're changing format, sometimes we're changing macronutrients, we're changing moisture content, maybe you're cooking or not cooking something. So there's going to be some pretty large changes in the microbiome there. From an individual pet perspective, it's a decent approach to try if whatever format you're working is not working for whatever format you're feeding is not working for your pet.
And I would add there too, right? So thinking about, again, we've talked about redundancy, we've talked about adaptability of the microbiome to respond to different food formats and looking at, okay, from a macronutrient profile, starch changes versus other diets, protein levels versus other diets. But then there's a step deeper that we need to think about, right? Even within certain macronutrient profiles or certain macronutrient classes, right? If I'm looking at starch and I'm looking at a starch that might not be gelatinized as well or cooked as well, is that making it further down the intestine and then going into the large intestine where it's a ready available food stuff for that microbiome? Protein as well is worth consideration, but it's specifically looking at digestibility of protein. If that protein's not utilized and used up in the small intestine and makes it into the large intestine, then we start to get some putrefaction, and the microbiome will respond appropriately to the feed stuff that it sees, right? So if I'm getting quite a large load of protein into the large intestine, then do I start driving some of those putrefactive microbiome that are then causing me greater stool odor, more phenols, more indoles coming from that protein breakdown. So even within macronutrient classes, it's important to consider quality of that macronutrient profile.
We had another question. We went a little deeper into specific ingredients and whether a raw ingredient such as tripe, which may have beneficial bacteria on it, maybe carrying beneficial bacteria. Can that ingredient be substantiated with microbiome claims if something like tripe that might already have the bacteria on there?
If the benefit that you're hoping to derive is coming from live bacteria on or in the food, I think it's really important to assess, are those bacteria surviving the stomach environment? That's really critical. That's a huge function of the stomach biologically, right? Is to kill a lot of these bacteria that are on these non-sterile foods that we eat so that they're not, to killing pathogens so that things are not getting into the gut. So that would be probably the number one thing that I would want to know about an ingredient like that if you're, again, hoping to derive that benefit from a live bacteria.
You can certainly characterize the microbiome profile of a raw material like that, but in the end, 2.5, 2.8 pH is an equalizer there. Thank God. Otherwise, we'd be sick all the time.
Yeah. And not to say though that the bacteria maybe even necessarily needs to be alive to produce that benefit, depending on what the benefit is. There's really interesting work taking the same strain of microbe and giving it live and giving it killed as a postbiotic, and the benefits are very similar. They're highly comparable. So you may not even need to do that, but it again, it depends on how you think that those bacteria are exerting that benefit.
The whole idea of the stomach is to help kill off some of these things.
Yes.
You get them to get in there. All right. Those were some great questions, and I'm getting pretty close to the top of the hour here. I was just wondering if either of you had any final thoughts and like to share those with us.
Yeah, sure. I'll go. I just want to say thanks, Tim, for the opportunity. Love the questions. Really good questions from the audience. So really appreciate those. I think we're starting to have more understanding about the microbiome. I would leave us with, yeah, we're starting to peek under the hood. Lots more to discover, lots more for potential to influence the microbiome, and excited to be in the space. So thanks.
Yeah, I'll echo what Mark is saying. Thank you so much for having us. It's been great, and it's been really fun to see the level of interest and everybody's great questions. I'm so thrilled that it's on people's mind, right? And that people are starting to appreciate the microbiome and all the things that it can do and all the ways that it affects pet health. So it's a field that I'm obviously really excited about. Looking forward to seeing where we can go with it.
Yeah, and thank you both for joining us. It was very educational for me as well, a very complex topic. You do have a lot of research opportunities out there. It's very interesting. Thank you both.
Thank you.
And also a thank you to our audience. We greatly appreciate your feedback on today's chat. You should now be seeing a QR code. If you scan that QR code, you will be able to access a survey, which we will also send you in a link in a survey email, trying to get your opinion on what you thought of the show. I'd like to give a great big thank you to Cargill for sponsoring this edition of Ask the Pet Food Pro. Please watch for more Ask the Pet Food Pro chats brought to you by Pet Food Industry and Pet Food Forum. I'm Tim Wall, Senior Reporter for Pet Food Industry. Thanks again to Dr. Jessica Jarrett, R&D Principal Scientist, and Dr. Mark Franklin, Global Technical Lead, both for Cargill. Also, great big thanks to the Watt Global Media team, who is making this possible behind the scenes. Thank you to our international audience for joining us. I hope you all enjoy the rest of your day or evening, depending on which hemisphere is your home. Until next time on Ask the Pet Food Pro, goodbye.